Hello. My topic will focus on the process in conducting a synthesis-based research. An example of this is in the design, synthesis, and characterization of biomimetic compounds. So one of my research interests is on biomimetic studies because I want to understand the mechanism of action at the active site or recognition site of enzymes or catalysts through its interaction with the substrate, activation of small molecules, conformational and other structural changes at the coordination sphere, and redox changes at the metal center. This has an impact on potential applications to synthetic chemistry, industry, medicine, and biology. So how do we go about it? Well, we synthesize model compounds by focusing on relevant functional groups, the size and shape of the compounds, and the simplicity or complexity of the model compounds. As an example, let me talk about bleomycin, particularly the iron 2 bleomycin complex, which is known to be a mononuclear non-heme complex that activates oxygen and promotes DNA cleavage. It is also known to undergo other oxidation reactions, such as epoxidation, hydroxylation, and demethylation. Now, it is known that oxygen coordinates with the iron 2 bleomycin and is transformed into hydroperoxo-iron 3 bleomycin complex, which is responsible for the oxidation of various substrates. It is also known that the arm, which coordinates at the axial position, has an effect on the redox properties of the catalyst. So we want to know if we change the functional group of the arm, how will it affect the chemistry of the catalyst? Now, in order to prepare a model compound, we simplify the design of the original compound by taking into account only the important functional groups of the complex. We also need to consider the simplicity of the synthesis. Thus, we prefer to make a more symmetrical molecule. Now, this model compound can then be prepared according to a previously published method by Karn and Bush. So our focus for today will be on the design and synthesis of nickel-2 complexes. Now, in their work, a diacetylpyridine and a dipodylamine compound are reacted in the presence of nickel perchlorate as metal template, forming a macrocyclic complex. Now, the imine groups are then reduced to form a more stable amine bond. The procedure is then reproduced by Herrera and others by using a tripodylamine compound as the starting material. Now, from both works, several pyridine macrocycles were synthesized with varying pendant arms, hydrogen bonding features, and acid-base properties. At that time, the known compounds are the following. Now, we want to add more macrocycles in the family, which includes those with an alkylated amino group, an amido group, and a carboxylic acid group. Now, the acylation of the amine arm is facilitated by a nucleophilic substitution of acetic anhydride in the presence of a base. Thus, I believe that alkylation is also possible by a simple SN2 reaction with an alkyl halide. Unfortunately, that did not proceed as expected. Okay? So it seemed that the SN2 reaction works very well, that a quaternary ammonium product is formed according to mass spectroscopy, even if an equivalent amount of methyl iodide was used. Another strategy involves a shift-based formation followed by reduction of the imine bond. However, the expected product cannot be obtained. So I tried a milder reducing agent such as sodium cyanoborohydride instead of sodium borohydride, and it worked perfectly, okay, to form the alkylated amine compound. Now, this teaches us an important lesson. Some reactions are good only on paper, but not necessarily in practice. Now, when you are doing synthesis, mass spectroscopy is a very useful tool or technique to check if you have produced the right compound. Sometimes you don't even need to finish the workup procedure to check the presence or absence of your target compound. You can get a sample from the reaction vessel and run mass spec right away. Introducing a carboxylic acid arm was initially thought to be a simple SN2 reaction between the amino group and the ethyl bromoacetate followed by hydrolysis. To direct the alkylation towards the middle nitrogen, I started the reaction using the oxidized form of the nickel complex. That way, only the more nucleophilic amine group will react with ethyl bromoacetate, but it failed. It also did not work well with the reduced nickel compound. So I tried experimenting with the uncomplex macrocycle, but it formed multiple alkylated products as shown in the mass spectrum. So based on this set of experiments, we can say that the nickel ion prevents alkylation of the NH groups. Well, presumably because the lone pairs of nitrogen are no longer available or nucleophilic because the lone pairs are bonded directly to nickel ion. And so I tried a different strategy. By retrosynthesis, we know that the macrocycle is formed by the reaction of diacetylpyridine and triamine compound. Therefore, we simply need to synthesize this modified amine compound. To do that, I use BOC-ON as the protecting group so that a reaction with ethyl bromoacetate will lead to the alkylation at the middle nitrogen. After deprotection, the N-alkylated compound was reacted with diacetylpyridine in the usual manner. Unfortunately, the macrocyclic compound did not form. Could it be that the arm is causing a steric hindrance or steric strain towards formation of the macrocycle? Now, to test this, I used ethyl bromopropanoate as the alkylating agent. However, again, the macrocycle did not form. Fortunately, I remember another interesting reaction known as the Asa-Michael addition, where a nucleophile reacts with an alpha-beta carbonyl compound to form a nitrogen-carbon bond. So here, the macrocycle is reacted with acrylic acid. The reaction can be monitored by TLC and later confirmed by mass spec. You will notice that the reaction mixture contains both the unreacted macrocycle and the target compound. So we need to think of a clever way to separate them. Now, one way to separate the two is to convert the carboxylic acid into a salt and perform solvent-solvent extraction. The starting material will be removed by chloroform from the aqueous solution containing the product. Then finally, nickel is introduced into the macrocycle. Formation of the complex is confirmed by mass spectroscopy. The same azamical addition is applied to introduce the amide arm. However, in this case, two alkylated products were formed based on TLC and mass spec. Fortunately, the mixture can be cleaned by complexation with nickel and adding diethyl ether to the mix. Through selective precipitation, the target nickel complex can be precipitated out of solution, leaving the impurities behind. Again, this can be verified by mass spectroscopy. Now, this diagram shows you all the synthesis steps to form the desired compounds. I also used NMR spectroscopy to verify the acquired products as shown here. And finally, structures of novel pyridine macrocycles were also confirmed using X-ray crystallography as shown here. So I hope you now have a grasp of the different skills, methods, and techniques required in conducting synthesis experiments. Thank you and have a nice day.